Welcome. It's my pleasure to introduce our speaker today. I'm Richard Hodis, Director of the National Institute on Aging, and happy to be welcoming you to the Florence Mahoney Lecture, the title of which will be Targeting Aging, Longevity, and Rejuvenation. In fact, Gladyshev is the speaker. But there are a couple of aspects of history, especially I want to attend to this afternoon. One is the background behind the named lecture, Florence Mahoney. So I had the pleasure of knowing Florence Mahoney. She died in 2002 at the age of 103, and she was really an extraordinary person who was largely responsible for the foundation of NIA, also some of the other institutes. She was a great politician, and she would hold court with influential politicians in her home in Georgetown, where a number of movies have been filmed as well. So, extraordinary woman. And we would meet before the selection of a speaker. She was insistent on hard science. She didn't want any of this soft science and nonsense. She wanted real hard science. And up to the age of 103, she was sharp enough. She would be delighted today by the choice of our speaker. And we'll get to another bit of history when we introduce that background. So before we get started, it won't be on screen, but for those of you who are interested in CME credit, the code is 57682. 57682, and in good days that would be displayed up here. But I think it's good for some time afterwards too, so if any of you want to come up and get it from us later, we can do that. In addition, those who are joining us by videocast, if you have questions, you can enter them under the live feedback icon, and we'll collect those, and they'll be read out and answered in the discussion period. For those of you who are here in person, we have microphones to do that live. It's also going to be a reception after the lecture, and want to make you all aware of that with some reasonably healthy and balanced refreshments. So to introduce our speaker today, Gladyshev is Professor of Medicine at Harvard, also an appointment at the Broad Institute, and Director of the Center for Redox Medicine in the Brigham. He's been a pioneer in the area of redox biology, and I'll elaborate on that a bit. He got his undergraduate degrees in Moscow. Then he came for postdoctoral fellowships here on campus, so he knows the place, or at least what it used to look like. And this is with NHLBI and NCI. But more to the point, I don't know how many of you notice when you walk by all the time the exhibit that's right up there on the Stadmans, the Stadman path. Earl and Terry Stadman, his first postdoc was in the Stadman lab. If you go up there and you look at a face mask attached to a tank, he's got the record for being second in place for the number of hours spent in there. This was to breathe air in a sealed nitrogen environment to look at anaerobic microbes. So this whole story of history couldn't be more fitting than for our speaker today. He's transformed the field, widely recognized with his contributions. Now an NIA grantee, a full professor. He's received the Eureka, NIH Pioneer, and Transformative Awards. His contributions have been many. And so without further ado and with that historical introduction, it's our great delight, Vadim, to welcome you here to speak today.
Thank you, Dr. Hodis. It's a great pleasure to be here and giving this Mahoney lecture to you, and thank you everybody for coming here. I have, as you mentioned, I've been here at NIH, and I have many good memories associated with this place. I've been working in the 90s as a postdoc, and I was working with Dolph Hatfield here in Building 37. He's just a wonderful human being. Yesterday had a dinner with him. He's long retired, but we have 125 papers together with him. And I've been working with Theresa Statman at NHLBI here in Building 3. As Dr. Hodis mentioned, there's a Statman Way. I encourage you after the lecture just go and look at this, because there's really a lot of history and it's an amazing place. So, but recently I've been, actually not recently, for maybe 20 years, I've been working on aging. And of course, I'm very grateful for NIA for all the support which allows us to study this. But the point is that once you start studying aging, it's hard to study anything else. It's just, the problem is so important that nothing really compares to it, I think, in biology and medicine. Because the idea is that most diseases that medicine studies are age-related diseases. And if we can target the aging process itself, we could delay the onset of all of these diseases at once. And this is why it's so appealing, but also it's incredibly difficult to do. And the first question that we need to ask is actually, what are we studying? Actually, when we say National Institute on Aging, but what is aging? Apparently, it's not easy to define, and there is a difference of opinion on that topic. Some people think that aging is like increased mortality rate, increased chance of dying with age. Some other people think it's age-related changes, or maybe a decreased loss of function. Or some people think it's just a process after development. Or other people think in terms of the biological age or maybe damage accumulation. And there's no like dominant idea. So it turns out that when we say aging, we use the same word to mean different things. But to me, it's the most critical question in the field because all experimental design is based on this conceptual understanding. So we need to think about this process first and then design experiments. And it doesn't happen often. So another way to illustrate this is this, in a very simplistic way. So we could study aging at the level of dying, mortality, but mortality is caused by diseases. We can study like morbidity, disease incidence, but diseases are typically caused by functional decline. We can study aging as a functional decline, but functional decline is caused by damage. So my thinking, and of course it may be right or wrong, as I said, there's no consensus in the field, that aging starts here. And then these are late manifestations of this process. And I was trained as a biochemist, and I often think about enzymes this way. And so let's consider an enzyme that converts a substrate to a product. There must be a gene for that enzyme that catalyzes the protein. Enzyme catalyzes some kind of reaction, but this enzyme is not perfect. It's composed of only 20 types of amino acids and has a flexible structure. So there is non-zero likelihood it will produce something else. Let's call it damage. So as a result, we have a gene with positive function. But as a result of that function, we have a negative consequence in the form of damage. And it applies to any enzyme, right? So we have, again, positive function, which is constant, and negative, which is cumulative, right? So if we use that enzyme early in life, unavoidably, we will have late deleterious consequences. We could extend this concept to other types of biological processes. And as a result, damage would come in the form of byproducts or mutations, epigenetic drift, all kinds of errors, imbalance in protein complexes or imbalance in cell composition of organs, and so on. All other known and unknown forms of damage. But the word damage is somewhat limiting because people typically think of damage as like molecular, some kind of damaging changes. But in our lab, we often use the word deleteriome. This encompasses all negative age-related changes that as a result of life. Yeah? You can think of, in terms of chemistry, as a cumulative damage at all levels of biological organization, or you could think of in terms of physics, like in terms of like entropy or disorder. So in that sense, to me, aging has a physical nature. It comes ultimately from physics, but manifests chemically. So then, life evolved to counteract that increase in entropy. And there are various strategies to do it. So for example, one strategy is simply to dilute the damage. So because there are so many variety of damage forms, the means of protection, they're always limiting. But cells don't need to know that what are the specific damage forms if they can dilute it. So for example, here in illustration, there is a cell, and the 2 damages, the cell grows, becomes 4, and then it divides 2 again, and then so on. And that's one strategy, to dilute entropy. Another strategy is like germline, for example. So when there's a succession of organisms through generations, that's also a way to deal with this increased entropy. As a result, the nature of aging is physical, I think. It manifests chemically, but counteracted biologically through life. And so we have this kind of concept in all of our research that we design in lab. And the actual research is organized like this. So we study longevity, for example, longevity across cell types, across mammals, because different mammals have species have different lifespans. And in fact, there's over 100-fold difference in lifespan across mammals, as well as longevity across interventions. This results in so-called signatures of longevity, which are omics patterns, typically omics, indicative of the potential of the system to live long. They may not change with age, but if we modify them, the organism may be shorter-lived or longer-lived. We also study aging, changes during aging, and here we quantify the aging process through biomarkers of aging. This may be epigenetic clocks, transcriptomic clocks, or proteomic clocks, or imaging clocks, organ-specific clocks, and so on. This results in actionable biomarkers of aging. So we actually need both signatures and biomarkers to study the aging process. And the third component is rejuvenation. This is something very new in the field, where we find that cells, or sometimes the entire organisms, can go to the lower biological age, the state of lower biological age. We call it rejuvenation, transition from an older state to a younger state. I would say that the aging process itself is irreversible. There's no way with all of these changes we could go that same road back. So aging is irreversible, but we could still target an organism or a cell or tissue where it could go to a slightly lower biological age. And so these strategies allow us to target aging and rejuvenation, identify interventions, which we are testing in mouse models primarily, and dietary pharmacological kind of genetic interventions. And I will give you a few examples of how we do it. So we first started with the genomes, and we actually sequenced many long-lived species. So the first one actually was this one. It's called naked mole rat. This paper we published already a long time ago, but I think it was the first paper that where a genome was sequenced with the goal of understanding longevity. Another one is called the Maryland mole rat, and this is a Brand's bat, also a very amazing animal. It's one of the smallest mammals, but lives more than 40 years. And this is the longest-lived mammal called bowhead whale. It lives more than 200 years. And what we find through these studies is that various species develop their own way to live long. So that the adaptations that they developed are not necessarily the same across these long-lived species. So for these reasons, we also try to identify common changes across various groups of animals. And our main focus is mammals. And so in this study, which was published almost 2 years ago now, we actually analyzed 40 species of mammals. They are shown here, in 3 organs, looking at the gene expression changes. And we find that some genes, they show increased or decreased expression with the change in longevity across species. But what is interesting, and 3 examples are shown here, if one gene, let's say, goes up with longevity in the brain, typically it also goes up in other organs. If it goes down, typically it goes down to other organs, which is initially was counterintuitive, actually. Why would it go, similarly in different organs? So, but the point is that in each organ we had like about 500 genes, maybe 500 to 1,000 genes that represent these longevity signatures, again, which is a very useful measure for us, measure of potential to live long. We also study longevity across interventions because now we know many ways to extend lifespan in model organisms. And I showed this slide from this paper published 5 years ago. And here we study interventions in mice that extend lifespan. And the data in the form of Spearman correlation matrix, and this is gene expression data. So kind of red means these gene expression changes are similarly induced by different longevity interventions. And blue is kind of in the negative way. So you can see one cluster, for example, we call it calorie restriction cluster. So calorie restriction induces gene expression changes which are similar to something like growth hormone receptor knockout or FGF21 overexpression or some genetic models like Ames, Little, Snell, methionine restriction, and so on. But another cluster, or maybe represented actually by one compound called rapamycin, is a well-known compound that extends lifespan. But interestingly, it induces changes which are quite different from the changes induced by calorie restriction cluster. So apparently there are many ways to modify the organism so that it lives long. And we don't know yet which of these strategies can be combined or maybe additive or antagonistic and so on. And this is currently being studied actively in the field. Now we have this longevity across species. longevity across interventions. And we also can add another dimension, which is age-related changes, aging itself. And now we could ask at the omics level, how are these three patterns related to one another? And this data is shown here from a paper. I have lab member Alex Stochastic, really amazing researcher in the lab who developed this study. And I will walk you through this slide. In blue are longevity across species. For example, brain across mammals, or kidney across mammals, and so on. And the changes cluster with one another. Yeah? So again, changes in different organs are similar that lead to longevity of the species, right? And this cluster is aging, like aging across muscle of mammals, brain of mammals, different tissues of human. Global means different tissues and species, and so on. They also cluster with one another. And this is longevity across interventions. Again, growth hormone receptor knockout, calorie restriction, rapamycin, acarbose, canagliflozin, and so on. And they also mostly cluster with one another. But they form three different clusters. And if you compare the clusters, what we find is quite striking to me. First, there is a slight positive correlation between longevity across species and aging, which on the surface means that longer-lived species are older. This makes no sense. Why would longer-lived species like humans be older, for example, than mice? But our interpretation is actually quite different. So when organisms age, they accumulate damage, but also this damage induces activation of protective systems. And apparently, these protective systems are also used by long-lived species. So longer-lived species, they are not older. They are just better protected. Another striking observation is that there is no correlation between longevity across species and longevity across interventions that we find in the lab. So whatever we do in the lab, dietary, pharmacological, genetic interventions, they are quite different from how nature extends lifespan. And to me, this represents an untapped potential to study longevity. The information that we could gain from analyzing longevity across species could be used maybe in the future for radical changes in lifespan, not something that we find in the lab which is quite marginal. I would say a typical intervention in mice is maybe on the order of 10 to 20%. We now continue actively working in this direction, trying to understand these principles of lifespan control. Now I will go to the next topic on biomarkers of aging. And to me, this is a true revolution in the field. This is something that happened about 10 years ago, and the first person is Steve Horvath, published his seminal paper in 2013. He calls it aging clocks. And we followed on that and developed a mouse clock. This was already 8 years ago. And I just use this to illustrate how it works. We sequenced the genome through reduced representation bisulfite sequencing of about 200 mice of different ages. And out of millions of CpG sites, identified a few, like in this particular case, like 90 CpG sites, that consistently change with age, increase or decrease methylation. And together, they represent a mathematical formula with each site has a particular weight in the model where we could accurately predict the age of the mice. This is the actual age of mice in months, and this is the predicted age. And you can see there is very good correspondence. So we can accurately predict the age of the mice. These four groups of mice are mice that have been subjected to calorie restriction. And CR is known to extend lifespan. You can see that any age, these mice are literally younger based on this clock. So this clock is very useful to identify interventions like this. So for example, if you would know nothing about calorie restriction, when the mice are 10 months old, we could measure and say, oh, yeah, they're younger. Maybe there is a potential to extend lifespan. And we don't need to wait until mice die. And this is the attraction of the using biomarkers in human clinical trials, because we can learn of the potential of certain interventions without waiting for late life outcomes. And on this part of the slide, it shows that even if we developed a clock based on the blood of mice and apply this to cell reprogramming, where we convert fibroblasts to induced pluripotent stem cells, the age based on this clock goes to 0. So of course, embryonic cells like iPS cells should be young cells, but it's really nice that even such a clock like a blood clock shows that the age is 0. So again, it extends the application of the clocks. But then the question is, what are the best biomarkers? Because the first biomarker that was developed was trained on chronological age. But in fact, you can measure aging in different ways. You could, of course, measure based on chronological age, train a model of chronological age. You can train a clock based on future mortality, and these are so-called second-generation clocks, like a Grimm age or Pheno age. You can train a clock based on the rate of change. That's another clock called Dunedin pace. And you can basically measure aging in very different ways. And to this day, it's still unclear what's the best way to measure. It goes back to the main problem that I posed earlier in my talk on the nature of aging. Because we view aging in different ways, and therefore it can be quantified in different ways. So for this reason, our lab and many other labs started a so-called Biomarker of Aging Consortium. And the leading person here is Vadim N, who's an instructor in the lab who started this whole thing and kind of brought together the community, as well as Jesse Paganik also. And this is the first paper of this group, kind of positional paper where we defined what is biological age, what is aging, what's the intervention, what's the approaches to address that. Field becomes very popular. For example, we had a conference last fall, and there was 450 attendees from 30 countries. Initially, it's kind of a new field, and it's just growing exponentially. It's really amazing. And so much talent, so many young people. People are coming from all kinds of disciplines. At the meeting, we had a challenge, a biomarker challenge, where anybody could compete. We collected data for 500 people from our Mass General Brigham Hospital patients and DNA methylation, and asked anybody, okay, tell us what's the age. Tell us how long are they going to live, because about 15% of these people died. We had samples from about 15 years ago. And this is how it works. These are the three winners. They go by nicknames. And this is the actual age of the patients from our hospital, and this is the predicted age. See how well they predict. This is the known clocks, and these are all new clocks from that competition. They all outcompeted the existing clocks. It's really amazing how, again, rapidly things developed. And mean absolute error is almost 2 years now. With the accuracy of almost just slightly above 2 years, age can be predicted of people based on the blood analysis. And I will show you a few examples of how we use the clocks currently in biomarkers. So, for example, in this study, which is done by Jesse Paganik, he's also an instructor in the lab and also has his own independent program, where we studied how biological age changes in response to stress. And he described several types of severe stress, like major surgery, severe COVID-19, pregnancy, and parabiosis. In each case, he found that under condition of stress, biological age increases. And once the stress is relieved, it goes back to normal. Well, we cannot say exactly to normal, but at least it's decreased. So, for example, in pregnancy, we find that women are biologically older in the 3rd trimester. And postpartum, biological age is decreased. And this nicely corresponds to what is known about like gestational diabetes, heart disease, mortality change during this period, which is also increased. And now we have many other models that kind of show this consistent pattern. Another example I'd like to show you from a study we published last year in Nature Aging. This was led by Albert N, who is, at that time was graduate student. Actually, he defended his thesis last week. He's now Dr. Ng. And he developed the first causal clock of aging. Why causality is important? Because when aging happens, things change, right? And damage is accumulated, but also, as I mentioned, protection against damage also changes with age. And when we build clocks trained for chronological age, we unavoidably incorporate both damaging changes and adaptive changes, kind of responses. The responses are good, but they also become part of the clock. And if we randomly build a clock and it happens to be based on these adaptive changes, this will be very misleading if we apply interventions. As a result, Albert developed an approach based on Mendelian randomization, developing clocks based on damaging changes and adaptive changes. And we call them damage clock and adapt age clock. You can see both of them can predict changes with age. But when we apply these clocks to interventions, they show radically different changes. So for example, here is reprogramming from fibroblast to iPS cells. And damaging clock shows rejuvenation. But adapt age does not show rejuvenation, which makes sense. You target adaptive changes, they may or may not be rejuvenated. So again, it suggests that further work is needed in order to develop biomarkers of aging. It's a revolution. It's really amazing what we have, but the many things are still unknown. And we need the effort of the entire community to do it. Another example I'd like to give is from another paper in Nature Aging last year. It was done Andrei Tarhov, who is a former postdoctoral fellow, now works at Retro company. And here we tried to understand the relationship between the regulatory changes and stochastic changes. Because during aging, of course, there are stochastic changes I expected. Mutations here and there, the epimutation and so on, or byproduct. But also maybe some changes may be kind of deterministic, in a way deterministic. Yeah? We call them regulatory. And here ask a question, which one actually wins, like with age? So is it like if you look at the youthful state during aging, is it like a co-regulated changes like this? If one changes, another one predictably changes. Or it's stochastic changes like this, random changes. And he found that many changes are deterministic, like regulatory during aging. But also many are stochastic. So again, we have this duality when we cannot really clearly separate the stochastic and deterministic nature of aging. This was about, and by the way, he has done it by analyzing single-cell DNA methylation versus bulk DNA methylation. Now I'd like to switch to another type of biomarker. We call it transcriptomic clock, and this was developed by Alex Stochastic. And here he used an amazing data set of almost 5,000 samples in mice, rats, and then later in humans as well, which represent various models of shorter and longer life, like genetic manipulations, like dietary, pharmacological, that kind of reduce or increase lifespan. And he developed a clock. This is a chronological clock based on the transcriptome. And you can see in blue is training set, in red the test set. You can see it works pretty well. In predicting chronological age, it's a multi-tissue clock. Based on the RNA-seq, we can predict age in different tissues. And because we had this longevity data, he was able to predict also develop a mortality clock, which quantifies time to death. And this is mortality clock. You see there is very good correspondence about actual and predicted mortality. And this is how, just one illustration of the usefulness of this clock. We apply this to interventions which are already known to extend lifespan or decrease lifespan. And this is control, and this is lifespan extending interventions. This is lifespan shortening interventions. You can see the clock can be used to distinguish these interventions nicely. And of course, in the future, it may be used with other interventions. This study is still unpublished, currently in submission, but we are very excited about this clock, and we currently apply to these many models. I mentioned this in a few talks, and we now have like several dozen collaborations on using this clock. This is another illustration of the application of this clock to models of age-related diseases. This is diseases in mice, like Alzheimer's disease model, like a chronic kidney disease, ischemic stroke, and so on. But in each case, we find that the diseased tissue in mice, based on the Stochastic clock, is older than the control tissue. So it's interesting that this really amazing relationship between diseases and aging, where disease is associated with increased biological age in that particular tissue. Now I'd like to tell you a little bit about rejuvenation. So far we discussed aging, it's represented by this water kind of going from the pipe, aging in this direction. And we can slow down this process by interventions like calorie restriction, rapamycin, metformin, and so on. But what we also would like to do, we would like to take water from here and take it all the way to here, which means going from an older state to a younger state. Is this even possible? It's still debated in the field, and some people think rejuvenation is simply not possible. But I think by now we have a few examples. And one clear example is iPS cells. I already mentioned this to you. So when we convert somatic cells to embryonic cells, literally we convert them from older cells to younger cells. They are embryonic cells, they are young cells. Another example is in this study, we published in Nature Aging. This is Bohan Zhang. He was a graduate student at that point, now is a postdoc in the lab, and it's a close collaboration with Jim White at Duke. And we used a model of a heterochronic parabiosis, where we connect young and old mice together for 3 months, and we follow them, what happens after 3 months, and then we separate and follow them for another 2 months, and then later until they die. And what we find that after this process, the old mouse is rejuvenated, and it lives longer compared to control. At the level of epigenetic clock, shown here, but actually we have other clocks as well, we find that these mice are literally rejuvenated. This is the situation 3 months after they are connected, and this situation is 2 months after they've been disconnected. The effect remains there. So they are still younger than controls, and eventually they live longer. Although I should mention that the change in biological age is much stronger than the effect of longevity. It seems like this time the effect diminishes. I mean, it still remains a little bit and that they still live longer, but the strongest effect is really at the time when they are connected. And over time, again, it decreases. But anyway, this represents another example of rejuvenation, and we also actively study this mechanistically. And the third example I'd like to show is actually currently the only known natural process of rejuvenation. And we discovered this a few years ago. This was a conceptual paper 4 years ago, and then we had a few experimental papers in mice, cells, and so on. And this was done by Csaba Kiripesi and Alex Trapp. Csaba is now a PI in Hungary, in his native country, and Alex leads bioinformatics at a biotech company. And what we find is that when at the level of the zygote, which is a fertilized egg, biological age is very low, but it's not zero. And initially, it kind of stays the same, and then about in the 3rd week of life, in human life, in mice, of course, a little bit earlier, there is a reduction in biological age. And the lowest biological age is found here. We call it ground zero. And from here, from this point, we could detect an increased biological age. We think aging begins right here at the ground zero level. And this is, in development, corresponds to the stage of gastrulation, approximately gastrulation. So again, we observed this in multiple situations. We developed, we see this at the level of epigenetic clock, like a bulk clock. We also have, I didn't include here, but Alex also developed a single cell epigenetic clock. And it's a probabilistic clock. It's quite different. It also shows rejuvenation, and transcriptomic clock shows rejuvenation. And even conceptually, we know that the germline ages slightly. For example, we know that the sperm from older men is older than sperm from younger men. So there must be a process of rejuvenation somewhere, but it was not known. But I think this happens here. Mechanistically, we don't understand, but we are very excited about this finding because, again, so it's a natural process of rejuvenation. Happens universally, appears to be universally across mammals and vertebrates. And if you understand mechanistically the nature of this process, perhaps in the future you could apply it to other systems, including somatic cells. I'll show you a couple of examples of how we study this. This is transcriptomic clock, and we apply this at a single cell level in mouse embryo from days 6.5 to 8.5 post-conception. And you can see there is a nice reduction in biological age. The question is, of course, at that point there are many cells. So which cells are rejuvenated and which are not? And we could apply our clock, and we can see that essentially all cell types are rejuvenated. They go to younger state at this point, with the exception of extraembryonic tissues. So we have a few examples of extraembryonic tissues, including the cells which later become placenta, which are not rejuvenated. They're still young, but not rejuvenated, whereas like cells that become an embryo itself are rejuvenated. And Now I'd like to tell you a little bit about another omics level to quantify biological age, and this is based on proteomics. And this is done by Ludger Gemini, who's a very talented postdoctoral fellow in the lab. Using UK Biobank data, he developed a clock that predicts biological age based on the plasma proteome. This is the actual age and predicted age and colors the men and women. This is the clock trained on chronological age, and this is a clock trained on time to death. Also works really well. But we've been thinking that we always discuss biological age of individuals. Like when we look at a person and say, oh, how old is this person? Or maybe 50 or 30 or 90. But actually, people are complex organisms. And their cells and tissues might age with slightly different rates. So what we really need to think is not just the biological age of a person, but biological age of different tissues and cells. And that's the paper we published on that. And I would mention that it was following another paper. It's an amazing paper from the lab of Tony Viscaray from Stanford, who developed this, pioneered this approach of organ-specific aging. Basically, what we did, we studied UK biobank population, 40 to 70 years old, about 45,000 people, and we identified proteins in plasma that come from different organs. Let's say proteins are made in the brain based on gene expression, and we detect them in plasma. We infer that these are brain proteins, and if we follow how they change with age, we could build a model of aging of the brain. And based on these plasma proteins, which can be easily accessible by drawing blood, we can predict the age of the brain. Yeah? And so this way we are able to predict the age of different cells and tissues in the body based on the plasma proteome profile. And when we develop the clock, chronological and also mortality clock, we can analyze like organ aging within the same individual. And these are residuals which are shown here for this organ aging. So now we have like clock for the liver, for the brain, artery, conventional, whatever, intestinal clock, kidney clock, based on the plasma proteome profile. Yeah? And we ask a question, how like residuals are related. So they're correlated with one another in chronological and also in mortality clock, which means that if somebody has older brain, typically is also like kidney is older. And so it's expected. Like organs like show correlation. But also, we find that there are significant deviations. And maybe the 2% of people, maybe they are more older in the kidney, and 2% might be older in the brain, or 2% more older in the heart, and so on. So, in fact, when we apply our organ-specific aging models, we find really amazing, we have amazing observations. For example, if you take a lung-specific model, and we look at diseases, the first disease that is predicted by the lung-specific aging clock based on the plasma proteome is COPD, a lung disease. If you take the liver-specific clock, liver cirrhosis comes first. So our organ-specific clocks trained on mortality predict age-related diseases in these people. Now we could relate these organ aging clocks with diseases. Again, so we find, for example, let's look people with dementia. Blue is young, red is old. You can see that in many of their tissues, they are quite young, but they are old in the brain. Like people with COPD, they are old in the lung, and so on. And we also have this kind of large metabolic cluster, we cannot clearly separate like kidney, liver, heart, and so on diseases because they influence one another. Yeah? So now that we have these organ-specific aging clocks, we realize that first organs age with slightly different rates, and this accelerated aging in an organ results in a disease in that organ. Yeah? We could, instead of looking for interventions that universally used in humans, we should look for interventions that target particular organs. For example, if somebody is old in the brain, maybe Alzheimer's disease, future Alzheimer's disease, we need to target the brain specifically rather than universally. I think there will be no universal intervention. Nothing will work universally for everybody. Calorie restriction is very useful, but we need to know who can benefit from it, because not everybody will benefit from it. And I will show you a couple of examples of why I think so. For example, let's look at the association of various environmental factors with organ-specific aging. like this organ ages, and this is smoking. So smoking is bad, right? So, but which organs are affected by smoking? And we find that actually most of the organs are affected. They become older by smoking. Let's look at drinking. Drinking is also, alcohol consumption is considered bad, unhealthy, but the effect is not as straightforward as in the case of smoking. So, for example, kidney age is increased, intestinal age is increased, but arterial age is not. Actually, if anything, it might be rejuvenated by alcohol. So, depending on who ages more in the kidney or intestine or like in the artery, alcohol consumption would have widely different, radically different effects. Just another example, just to illustrate this point. Let's consider arterial clock. Again, plasma proteome. We take proteins which are secreted by artery, and we tell the age of the artery based on the plasma proteome. And we look associations with like food, in this case food, like yogurt and wine. So wine for the artery seems good. Yogurt is bad. But if you look at intestinal aging clock, it's opposite. So now wine is bad, yogurt is good. So depending on who ages more in one organ or another, a different intervention would need to be used. The intervention needs to be personalized. So what does it mean? First, we observe asynchrony in organ aging across organisms. And of course, I'll tell you about the proteomic-based measures. But we are trying to develop additional measures of organ-specific aging. And of course, not only in my lab, but it's an active area of research. I think we need to think of targeting organismal weak links. If somebody ages more in one particular system, let's target that, because that system will result in a disease and the person will die from it, most likely. Need to think of personalized interventions. This is our model. Life begins here, aging begins here, remember ground zero, third week of life in humans, and then organism develops and ages from that point on, but unequally in different tissues. And in this particular case, just an example, heart ages more than other organs. But the heart is not simply older. Heart interacts with other organs because there's a crosstalk. So other organs which are younger pull it back. So without these organs, it would be even older. But now it's in a slightly older but also altered state. And we still don't know exactly how to describe that kind of situation. But what I can tell you is that to us, basically, we think of diseases as aging. So maybe it's an extreme kind of example, but to me there are no diseases. All we have is aging. So aging happens, and some organs, they are aged more. Not necessarily organs or systems, or some part of the organ, and that state we call a disease. So from that perspective, kind of diseases and aging are kind of can be unified. And it highlights the importance of the aging process in any kind of health-related issues. So I'd like to summarize what I was telling you today. So first, we discussed a little bit about the nature of aging. It was telling you that aging, to me, is the increased deleteriome. It begins at gastrulation, or ground zero, and it's not an adaptive process. It's like a gravity, basically, in physics. It has physical inevitability, manifests chemically, and mitigated biologically. We also discussed longevity signatures, which are omics patterns that predict the potential of the system to live long, discussed across species, cells, and across interventions. I highlighted differences between species longevity and longevity induced by known interventions, which highlight, again, so indicates the potential of this to develop new, radically lifespan extending interventions. And also a little bit discussed about the genomes of long-lived species. Also, I discussed a lot about aging biomarkers at the level of bulk, single cell, causal, stochastic, multi-tissue, this epigenetic, transcriptomic, and proteomic, but they are not limited to that. Of course, many labs, including my lab, we work on multi-omic biomarkers currently. Also, I highlighted reversible changes in the predicted biological age upon severe stress. And also we discussed the Biomarker of Aging Consortium and Biomarker Challenge, which is a community effort that we support. Another topic discussed was rejuvenation, and examples are cell reprogramming by Yamanaka factors. I didn't mention chemical, but we also last year published a paper on chemical reprogramming, but a cocktail of 7 compounds where we're able to reduce biological age. Also, this example of natural rejuvenation, culminating in ground zero, and as well as heterochronic parabiosis. These are some of the examples where biological age can be reduced, even though the aging process is irreversible. And also, final part of my talk was on organ-specific aging. I told you about organ aging models based on chronological models, mortality-based models, and association with diseases and diet, where I think that accelerated biological age subsystem, kind of accelerated aging, is a disease, is represented by disease, and therefore this biological age as a measure can be used to target that particular process before it happens, preemptively. So diseases in that context can be viewed as pockets, non-autonomous aging. But ultimately, what we do, all we do, is try to understand aging. Try to understand, again, so there is no consensus. What I told you, it may be all completely wrong, I don't know. But it's such an important process because it affects our health. Like the mission of NIH depends on studying aging, in my mind. That's the most important question. And this is my lab. Various people have been involved in this work, as well as previous lab members. We have many collaborators. I would like particularly mention Luigi Ferrucci from the NIH, who's a great colleague, and what happened yesterday, I hope, is a mistake. And of course, our funding, all of our funding, most of our funding is from NIH. I'm incredibly grateful for NIH and NIH for allowing us to do this work. We are very passionate about it and hope to continue making progress in the field, as well as other funders. And with this, I would like to thank you for your attention.
Yeah, Phil, please start. Yeah, lots of questions online too, but please start here.
That was a wonderful talk. Can you hazard a guess on that original source of damage that initiates the aging process? What do you think about reactive oxygen species or inflammation as the upstream event that drives all this in all the organs?
My opinion is that there is no a single cause. So much of the field studies particular damage types, like you mentioned reactive oxygen species, and I come from the redox biology field. That's my connection to aging, in fact. But 10 years ago, I published a paper with the title, "The Free Radical Theory of Aging is Dead." Literally, that was the title. To highlight the point is that we should not limit ourselves to individual damage types. So there is no one most important damage type. Damage comes from the totality of biology that happens. Every cellular function has positive effects, the function itself, like genetically encoded typically, but also negative effect, which is cumulative. And if we try to isolate one component, it's already going to be misleading. So it's a totality of damage. That's why we call it the deleteriome. Yeah, and it makes it very difficult to study because we trained as reductionists. Like out of complex picture, identify a component, study it, and through it explain the process. But it doesn't work in the case of aging, in my opinion.
Yeah, I have two questions. The first, going back to the beginning of your talk, what's the evolutionary advantage of long life? So if you're a small rodent, let's say, that can mate when you're a few weeks or a few months old, what's the advantage of living to the age of 40? And my second question has to do with parabiosis. So I've been hearing people talk about this now for a number of years. So there must be something in young blood that can be used to reverse aging in older animals or older people. And yet I haven't heard anybody talk about a substance, a chemical, a protein in young blood that has that effect. So what's the reason for that?
Okay, thank you. That's great questions. Now I forgot the first question.
Oh, yes, the evolutionary advantage.
Oh, evolutionary.
Yes.
Okay, so the animals adapt to to the environmental niche, and therefore sometimes it's beneficial, like to reproduce very rapidly, like in the case of mice and rats, and sometimes just there's advantage to live longer and invest more into like a few like progeny, but kind of protect them really well. It's really it's an adaptive process, right? So, but the adaptation is for like the fitness in that particular environmental niche, and kind of longevity comes separate. So I'd like to make this distinguish aging from longevity. Again, I already tried to a couple of times. Aging is, to me, is a spontaneous process, non-adaptive. Longevity is adaptive. Yeah. So, and the parabiosis. You're right. So there have been many studies, and we still don't understand mechanistically how it happens, because it's a complex model. So when we connect the mice, it's not simply like rejuvenating factors in the blood that go to the old animals, but also all the animals have access to the younger organs. And it's very difficult to separate this. So to me, it's most likely a multifactorial process where like maybe damage dilution, removal of damage through younger organs, possibly rejuvenating factors, and once you try to separate, isolate, you lose the effect. So in our hands, we cannot recapitulate this effect by young blood cells, for example. So we don't see rejuvenation. Of course, we look at rejuvenation of our other organs. We cannot look at the blood itself. We typically look at like, is liver rejuvenated? So, and we don't observe this effect. But parabiosis works, but once we go like individual models, somehow we lose the effect. It's not an easy question.
Okay, thank you.
Vadim, first of all, thank you for a very stimulating lecture, and it's like more you know, you're opening a Pandora box, so all kinds of interesting things pop up. My question is very narrow. There has been a lot of attention in the last few years on exercise as a resetting a metabolism that would affect many organs. Have you looked at some of the effects of exercise in your model, and if it resets some of the aging mechanism of individual organs, which would be of great interest?
Yeah, that's a great question. We haven't studied in my lab exercise. Of course, there are many other labs who study, and they do observe health benefits from exercise, of course, in humans and in mice. I haven't seen anybody looking at organ-specific. Actually, we did look a little bit in the UK Biobank. We can also predict the age based on like imaging data, for example, like a DEXA scan. And in this particular case, like we find that severe exercise actually accelerates aging of the bone, even though it rejuvenates, helps to slow down aging of internal organs. So there is a kind of contrasting effects that we observe. But it's a preliminary data. It's not yet published.
So, Vadim, taking off on this question, there are resources around, reagents, specimens, for example, from exercise interventions, like in MotorPack, with the ability to look, even if it's just serum, at, for example, exosomes or particles which can be attributed to various organs and tissues. Is this potentially a rich source of information about an organ-specific effects over aging and with intervention that's there for the analysis?
Yeah, definitely. So you're right. So these methods are it's very new, right? So just published and limited to some data sets. Like in our case, it's a proteomics-based O-link, and Tony Viscara is primarily a somologic. But you're right. So with these and additional approaches, we should be able to go organ-specific in an organ-specific manner and study all of these effects, including exercise. Yeah, you're right. Yeah.
Hi. So I thought it was really interesting how certain interventions like caloric restriction or senolytics had a different like aging signature in terms of like aging reversal rather than just like how you age. And I was wondering if you've looked at how reprogramming or that rejuvenation stage during embryogenesis or parabiosis, how those aspects of reducing aging might look in that context.
We actively study this, but we don't have insights yet because we want to better define this rejuvenation states. And like if you noticed in my talk, like in the case when I talking about reversibility in biological age, I didn't use the word rejuvenation because like in this case, actually we don't know. If we artificially increase it and then like decrease to the normal state, is this rejuvenation or not? I was referring to rejuvenation when we can go from the normal state to like younger state. Then we say rejuvenation, but biological age fluctuates, like seasonally through stress, whatever, and we need to better understand those situations. Yeah. But it's an excellent question, and hopefully in the future we'll know more about it.
Hi. So I thought the mention of organ-specific aging was super interesting. So I understand that you've done some work on how organs age asynchronously. I was wondering if you've done or conceptualized kind of interactive studies, so kind of understanding how aging of the lungs would correlate with aging of the brain, or aging of the pancreas correlate with aging of the eye, or skin with the liver, etc.
Yeah, again, so this is all very new, and in one slide I showed that there is a correlation. If you look at the population level, there is correlation in one organ, aging one organ corresponds to aging of another organ in general, but there is outliers. Sometimes it's difficult to relate various organs. Like, for example, in the plasma proteome-based organ-specific logs, we have access to maybe 8 or 9 organs and tissues, right? So for others, just not enough data. We need better data set. But like eye, for example, would not be part of it. So we try to look at the eye through retinal imaging, and also, but it's again, so we're working on it, but we are not able to relate yet to internal organs.
Thank you.
Hi, so I was curious about your models on organ-specific aging as well. What did the data look like that you built those models on? For example, how are you able to build a model that could predict, for example, how yogurt affects intestinal aging?
Okay. First, how we develop the clock. So this is an O-link data set in UK Biobank. It's 50,000 people. And the data set has about 3,000 proteins, a quantification of 3,000 proteins. We look at the organ-specific expression of genes based on gene expression, because we cannot look at the proteins. Proteins, if they are like in the organ, in the plasma, it will be difficult to interpret. But gene expression, if it's only expressed in one particular organ and detected in the plasma, we can say, okay, protein comes from that organ. So now we focus on only that subset for a particular organ, and sometimes it could be like 20 or 50 or 100 proteins which are unique to a particular organ. And we built aging biomarkers based on that, right? Now, by profiling plasma, we can tell the age. And because we have other proteins for other organs for the same person, we can tell age of the brain, liver, kidney, and so on, intestinal artery. So now there's information about the diets for these people. Not only diets, but drugs, like professions, like lifestyles. We do this, all of this. And we can find associations of these kind of lifestyle changes with organ aging. So, you see?
So all the sort of dietary data was self-reported?
It's in this particular case, it's self-reported. We also, we could quantify, we do various ways, but self-reported, but also we quantify the total amount of certain types of food based on the known characteristics. When people say what they eat, we could quantify based on calories and components of the food.
Got it. Thank you.
Hi, very interesting. So a question, so when you talked about the different organs aging at different speed in different people and different interventions like foods having different effect, does your body tell you what to eat depending on the signals that you get from your organs? For example, like your artery is aging faster and you have a craving for white wine, or your intestine, and then That's why you like the yogurt during these 2 years, for example, and then don't anymore. Does this happen in your opinion?
It may be. It may be the case. We haven't studied this. It would be very interesting to test. I agree. It's an important question.
I thought it was very interesting how you described cell division as a dilution of damage. And I was wondering, do you think the same concept on a macroscopic level is contributing to the effect of parabiosis, where it's diluting like damaged products in the blood? And then as a follow-up to that, do you ever test whether the young mice after parabiosis live for a shorter amount of time?
Yeah. So mechanistically, we don't fully understand heterochronic parabiosis, but I think damage dilution is definitely one of the factors in changing the biological age. In terms of the old mice, we actually do have the data. In fact, we published a paper on that. And we found that after 3 months, younger mice, they dramatically increase their biological age, almost like double. And then after we separate them, 2 months later, the biological age goes back to almost normal. So it's a reversible change. It seems like under stress, they are not able to deal with all this damage that comes from the older mice. But after the mice are separated, slowly they are able to recover and kind of remove that damage. And therefore, biological age is decreased. The accuracy of the clocks is insufficient to tell whether they're exactly the same as like normal or slightly above. This we are not sure. But it definitely shows reversibility. Mm-hmm.
Thank you.
Mm-hmm.
Thank you for a fascinating talk, a tremendous body of work.
Thank you.
My question has to do with physiologic stress-related changes in aging. And you mentioned as an example pregnancy that accelerates aging in the third trimester. And then there is a reversal of the phenomenon in the postpartum period. And my question is there a mechanistic explanation for this reversal that occurs in the postpartum period? Mm-hmm. And the second related question is you map the pattern of aging of different organs. And my question is, we have an organ that has a timetable that is the placenta. Do you know if there is aging of the placenta?
Let me go first with the placenta, because we actually did test it, because I was thinking that placenta has exactly as you say, has a particular time span and therefore might be older. But when we test it, we still find it young. Somehow it ages, but doesn't age as I thought it would age. It would age more like than the embryo, but it ages about the same for some reason. Mm-hmm.
So it may be that there is a process that is not measurable with the tools that we have.
It's possible.
But perhaps it's the way the question is framed. There is physiologic pregnancy and then complicated pregnancy. And then in the context of complications, let's say prematurity or preeclampsia, would that affect?
This premature kind of pregnancy, we have a project on that. We study this. We don't have data yet, but that's exactly what, like early life adversity, basically we call early life adversity, how it affects biological age. But in terms of reversible changes during pregnancy, I think damage dilution is an easy kind of way to explain it. And there's a huge metabolic stress during pregnancy on women, right? And so they are not able to deal with all of the damage that is generated. But postpartum damage can be slowly removed. But sometimes I'm getting a question whether when they recover, the age would be even lower than normal. And the reason is because of the parabiosis with the fetus. And that's also a very interesting question. After we published our paper, we published a paper on cell metabolism about a year ago. There was another paper published later also in cell metabolism. They found that there might be a reduction even more significant. Our resolution of our clock wasn't good enough, but in their case was a better data set. And they found also that women who breastfed their babies, they also recovered faster. So it's interesting. So it's new, so it needs to be, of course, reproduced by other labs, but it seems like at least in the case of reversible changes during pregnancy, the two labs have similar data. Mm-hmm.
Maybe one more, Dr. Hodis, and then we'll finish up over here. She was there first,
Mm-hmm.
yeah.
Thanks for the talk. And my question is, the aging community seems to use that deviation from true prediction as a measure of rejuvenation? Like if we're predicting with less accuracy, if it's predicting younger. So my question, even if you have a causal clock and you're incorporating SMR, how do you know if there's really relevant biological context in rejuvenation?
Yeah, it's a very good question. So the concept of biomarkers of aging is very new, right? And it's possible that if you just use one individual biomarker, it might have misleading effects. So in any of the studies that we find, there is always a caveat. We cannot be 100% sure. That's true. So, but for example, in the case of like embryonic rejuvenation, that's why I mentioned we have different types of clock, completely different. Like bulk clocks, probabilistic clocks, transcriptomic clock, epigenetic clocks, they all show the same. And conceptually, we expect to see that as well. So then we say, okay, we are quite confident there is embryonic rejuvenation. Also for parabiosis, like we use all kinds of biomarkers, we observe kind of consistent effects, so we are quite confident. But in many other cases, we are not quite sure. I didn't mention these cases, but if you look in the literature, there's many examples of rejuvenation. Sometimes it's hard to distinguish. Is it rejuvenation, slow in aging, or simply like effect of a particular biomarker?
How do we reconcile in terms of some senescent cells in aging, in disease, in cancer, they start to have this stemness factor, and they seem to predict younger, but they are still increasing morbidity.
Senescent cells younger?
Yeah, an increase in embryonic stem cell markers.
Embryonic stem cell markers in senescent cells.
I might be wrong.
Yeah.
And cancer cells.
Cancer stem cells. We're not focusing on cancer. I cannot be 100% sure. I just know that in cancer, sometimes in the cancers we observe reduction in biological age, maybe because cells like divide a lot and they damage. Sometimes it's increased biological age. So there's a lot of heterogeneity in the cancer. But I haven't seen data which would show that senescent cells are younger. I just haven't seen it. I've seen that senescent cells are older, but maybe I missed something. Yeah.
Thank you.
Yes, thank you first for a fantastic lecture. The overtime and no one's leaving. Apologies to those who were online and have their questions without time. We'll see what we can do about it. And just for those who didn't get the CME code beforehand, there it is, 57682. Still good. You can enter it now. And yes, please join us up on Stadman Way for the reception to follow. And thanks so much, Pat. Great talk.
Thank you very much.